All right, and we're back. We're gonna jump right into where we left off. Um, had to take a break, so we were right about here. I want to say maybe. Let's go up a little bit more. Ba-da-ba-ba-ba-da-bum. Oh boy, I don't remember any of this. Okay, I do remember this. The production of the B cell. Yep, I remember reading that. The whole VDJ. Yep, IgMs. How there's a different chain and different chances of things happening. Yep, yep, yep. The tyrosine kinase signaling. Yep, okay. Changes in characteristics of surface markers. Uh, hmm. Did we do any of this? Immune tolerance, central tolerance. Don't think we. Hmm. I don't think we actually talked about those. Definitely not. Alright, in that case, I think we ought to continue with the T cells. Let's review. Worst case, I read this twice, which is fine. Changes in characteristics, surface markers. B cell differentiation is also characterized by the development of a variety of important surface molecules. These include CD21, a complement receptor, and CD40, adhesion molecule required for later interaction with Th cells. Yeah, I don't think I've gotten that far. I think this is where we actually stopped off, so if that's the case, let's keep rolling. Central tolerance. Because assembly of the VJ and VDJ regions is random, B cell receptors will be produced that react with self-antigens. Further development and release of these autoreactive I lost my space there. Further development and release of these autoreactive cells into the circulation would result in a catastrophic immune attack against the individual's own tissue. One stage at which immune tolerance, suppression, or limitation of immune response against self-antigen occurs is the deletion of autoreactive B cells in the bone marrow, which is referred to as central tolerance, induction of tolerance within the central lymphoid organs. During the very early stages of formation of the BCR in the bone marrow, a large number of autoreactive B cells undergo apoptosis, clonal deletion, if exposed to self-antigen. This process of negative selection induces the death of more than 90% of developing B cells. Some autoreactive clones persist and must be controlled by other means in the lymphoid organs or peripheral tolerance. Okay, so at one stage or another, the immune intolerance suppresses the immune system against self-antigens. Then we have the central tolerance, which is the induction or tolerance within the central lymphoid organs. And then we have peripheral tolerance, in which some of the B cells or clones persist that need to be controlled by other means. Very well. In which case, we're looking for some T cell maturation. So, the central lymphoid organ, the jumping. The process of T cell generation of clonal diversity is similar to that for B cells. The primary lymphoid organ for T cell development is the thymus, which is an organ located near the heart. Lymphoid stem cells migrate to the thymus and enter the subcapsular region. As the cells journey through the thymic cortex to the medulla, they are instructed by interactions with various thymic cells, epithelial cells, macrophages, and dendritic cells. Thymic hormones, or thymosin, thymopoietin, thymostimulin, and others are produced by the thymic epithelium, and the cytokine IL-7 to undergo proliferation and progressive development of the characteristic of immunocompetent T cells. Changes include development in the T cell receptor complex and expression of characteristic surface molecules. The final immunocompetent T cells are released into the blood vessel and lymphatic vessels to establish residence in the secondary lymphoid organs to evade antigens. Radio. The production of the T cell receptor, which is one of the words we're going to be looking up here. Within the thymic cortex, the cells begin rearranging the variable region, genes necessary for forming the functional T cell receptor. The basic process of TCR, or T cell receptor rearrangement, is essentially identical with formation of the B cell receptor. And although the structure of the T cell receptor closely resembles the Fab portion of the antibody, the T cell receptor uses different genes than are used for antibodies. The most common T cell receptor contains alpha and beta chains, each of which have a variable region and constant region. We're going to talk about the variable and constant regions again. So, the variable regions of a T cell receptor alpha and beta chains also undergo somatic recombination. The alpha chain gene, chromosome 14, rearranges V and J regions, and the beta chain, chromosome 7, rearrange the V, D, and J regions. The somatic recombination of the alpha chain proceeds as described for the antibody light chain using the set of at least 70 V region genes and at least 50 J region genes. Okay. Thus, potentially having 3,500 different alpha chains may be generated. Right. I got a feeling this is very similar to what we did or read with the B cell. Okay, so the rearrangement of the beta chain occurs similarly as described for the antibody heavy chain rearrangement. The major difference, however, is the option of using two separate C gene regions, C beta 1 and C beta 2, each of which is adjacent to its own set of D and J gene regions. Somatic rearrangement of the beta chain gene segment uses at least 60 V gene regions and 2 C beta regions, each containing at least 1 D and 6 or 7 J gene regions. So as many as 720 different beta chains may be produced. As with the generation of antibody diversity, the primary mRNA for alpha and beta chains contains V region and C region exons with an intervening intron that is removed by RNA processing. Assembly of alpha and beta chains result in T cell rearrangement diversity that potentially represents 2.5 to 10 to the 6 antigenetic specificity. Right, a shit ton. Let's just say that. So for both alpha and beta chains, the V region genes encode the amino acid sequence that includes CDR1 and CDR2 and their appropriate FR regions. The J region contains information for CDR3 and FR4. The TCR beta chain D regions encode a short amino acid sequence found in CDR3 and greatly increases the diversity of the beta chain CDR3. As with somatic rearrangement of antibody light and heavy gene regions, the level of diversity, especially for the CDR3 regions, may be increased further by other factors. Imprecise joining increases the diversity of the CDR3 regions. For example, the sites of VJ and VDJ joining may shift slightly, resulting in an amino, though the alpha-beta TCR is a preferred antigen receptor. Some T cells, about 5% of the total T cell population, uses alternative genes, gamma and chromosome 7 and delta, chromosome 14 in the middle of an alpha chain gene. These cells with gamma delta TCRs appear to migrate to unique areas of the body. The epithelial areas in the skin, reproductive tract, intestines, respiratory tract, and have different and less well understood functions than the T cells with alpha beta TCRs. A functional TCR requires intracellular signaling molecules. Insertion of the TCR into the plasma membrane is associated with expression and association with TCR accessory molecules, collectively called CD3, that provide signaling to the nucleus after the binding of antigen to the TCR. CD3 is a marker for T cells that successfully form a TCR complex and are immunocompetent. Whew. Alright, so changes in characteristic surface markers. Differentiation of T cells in the thymus also result in changes in the variety of important surface molecules. Much of T cell development is controlled by hormones and cytokines in the thymus, and an early step in maturation is expression of the receptor for interleukin 7, which is a major cytokine that drives the differentiation process. Transit through the thymic cortex initiates expression of the molecule CD2 on the cell surface. CD2 is expressed on virtually every subpopulation of cells that have undergone development in the thymus and is thus a T cell marker. The developing T cell also begins making two other important surface proteins, CD4 and CD8, which are concurrently expressed on the developing cell's surface at this stage. CD4 plus and CD8 plus cells are often called double positive cells. After entering the medulla of the thymus, the double positive cells become singly positive. That is, some of the cells suppress production of the CD8 molecule and remain only CD4 plus, whereas others suppress CD4 production and remain CD8 plus. The phenotypic change to single positive cells is driven by exposure of CD4 plus and CD8 plus cells to MHC antigen expressed on cells of the thymus. The CD4 and CD8 molecules react specifically with MHC class II molecules or MHC class I molecules, respectively. For instance, the double positive cell initially comes into contact with MHC class II molecules. On the thymic cells, the T cells will suppress expression of the CD8 and become CD4 single positive. However, if the initial reactivity occurs between CD8 and MHC class I molecules, the cells will become CD8 single positive. Thus, this positive selection process results in two groups of immunocompetent cells with different functional characteristics. CD4 cells develop into T helper cells, or Th cells, in the clonal selection process, whereas CD8 cells become mediators of cell-mediated immunity and kill other cells directly, or TC cells. Approximately 60% of immunocompetent T cells in circulation express CD4 and 40% express CD8. Central tolerance. During the random rearrangement of VJ and VDJ genes to produce the T cell receptor, some combinations result in specificities that recognize self-antigens. If some of these autoreactive T cells were allowed to progress further in development and leave the thymus, a severe immunologic reaction against the individual's own tissues could result. One stage at which a tolerance for self-antigens is maintained is the deletion of autoreactive T cells in the thymus, which is referred to as central tolerance. A large spectrum of self-antigens is expressed on the surface of the thymic macrophages, dendritic cells, and especially the epithelial cells. If the developing T cells, or TCR, binds strongly with self-antigen, it will undergo apoptosis or clonal deletion. Although this process of negative selection induces more than 95% of the T cells to undergo apoptosis in the thymus, a limited number of autoreactive clones persist and must be controlled by other means in the peripheral lymphoid organs. So, peripheral tolerance discussed in this section titled Antigens and Immunogens. So, the induction of an immune clonal selection. Antigens initiate the second phase of the immune response, clonal selection. The process proceeds through 3 finely tuned sets of intracellular collaboration that results in the production of the effector cells, Th cells, plasma cells, Tc cells, and memory cells that provide long-term specific protection against infectious microorganisms. Step 1 is antigen processing and presentation. This is the selection component of clonal selection, in that the particular antigen is selected for maturing those B and T cells with related TCR and BCR specificities. Step 2 is induction of the population of Th cells. Immunocompetent CD4 T cells respond to present antigen and proliferation and differentiate into populations of effector Th cells. So Th1, Th2, Th17 cells. Step 3 is induction of immunocompetent B cells into plasma cells and immunocompetent CD8 T cells into Tc cells. This step requires the presence of antigen presentation and effector Th cells. Step 2 and 3 are dependent on highly regulated interactions among cells, APCs and immunocompetent Th cells in step 2, and APC effector Th cells and immunocompetent B or CD8 T cells in step 3. Successful intercellular collaboration depends on 3 complementary signaling events: antigen-specific recognition through the TCR complex, intercellular adhesion between specific cell surface, receptors and ligands, and secretion of and response to specific groups of cytokines. Without all of these signaling events, the protective immune response would not be produced. So we need all 3 of those to get a proper immune response. Clonal selection usually begins at birth and proceeds throughout the life of the individual as new antigens are encountered. Although it can begin as early as the 8th week of gestation in humans, if foreign antigens are encountered in utero, most commonly foreign antigens encountered in utero are related to the fetal infection during the last third of gestation. Right. So what does that have to do with that sentence in all reality? Most commonly, foreign antigens encountered in utero are related to the fetal infection during the last third of the gestation. Right, but what does that mean? Just that in the 3rd trimester, there was an infection? Okay. It doesn't explain how or why or what. Just most infections happen in the 3rd trimester. Got it. Secondary lymphoid organs. Most aspects of clonal selection begin in the secondary lymphoid organs, the spleen, lymph nodes, adenoids, tonsils, Peyer's patches, in the intestines, and the appendix. Immunocompetent lymphocytes enter the secondary lymphoid organs through the blood and enter specialized small veins called high endothelial venules, or HEVs, where they bind to the endothelium through the family of adhesion molecules. The lymphocytes migrate from the vessels into the lymphoid tissues, which contain B and T cell rich areas. B lymphocytes that encounter antigens in the secondary lymph organs usually undergo a process of differentiation and proliferation that result in the formation of specialized germinal centers in these organs. Fancy. Antigen processing and presentation. Most antigens do not react directly with T or B cells, but require processing and presentation in the appropriate fashion. This is the duty of APCs. Most cells have a capacity to present antigen to some degree, but dendritic cells, macrophages, and B lymphocytes are so efficient at antigen presentation that they are considered professional APCs. Each is responsible for the presentation of antigen of different types and from different sources. B cells can process soluble antigens and present them to Th cells that facilitate development of the humoral immune response. So, let's repeat that. B cells can process soluble antigens and present them to Th cells that facilitate the development of the humoral immune response. So are those considered B lymphocytes? Let's find out. Macrophages are effective APCs for the development of immune responses against antigens or antigenic components of infectious diseases. Bacteria, contamination of tissue by infectious microbes usually initiate an inflammatory response, and the infiltration of macrophages into the site. Macrophages are also very effective in presenting antigen to memory Th cells in order to initiate a rapid response to antigens that have been previously encountered by the immune system. The dendritic cells are perhaps the most effective in presenting antigen to naive immunocompetent Th cells. Dendritic cells develop from the bone marrow precursor cells, either of myeloid or lymphoid lineage. At least two populations of dendritic cells have been described. They migrate to the peripheral tissues, the skin, intestinal tract, and to secondary lymphoid organs. Immature dendritic cells at a site of inflammation function as phagocytes, and the process of phagocytosis can initiate differentiation and direct migration of dendritic cells to the secondary lymphoid organs, particularly the lymph nodes. Thus, dendritic cells can carry processed antigen from a site of inflammation to the T cell rich areas of the lymph nodes. Okie dokie. Additionally, infectious microbes or fragments of microbes may be drained by the lymphatic vessel to lymph nodes. The lymph nodes are extremely rich in dendritic cells and macrophages. Pathogens entering the bloodstream may be removed by phagocytic cells in the spleen and other lymphoid tissues. In either case, the phagocytic cells that engulf foreign microbes or their fragments are also responsible for processing antigens from the pathogen and displaying or presenting those antigens on the phagocyte's surface to neighboring lymphocytes in order to initiate the adaptive immune response that is specific to pathogens. Got it. Pathways of antigen processing and presentation. In general, the immune system responds to two types of antigens, exogenous and endogenous. Using infection as a model, exogenous antigens are carried on microorganisms that are trapped and killed by phagocytic cells. Therefore, they come from outside the cell. Endogenous antigens are synthesized within a cell. These include viral antigens, because viruses infect cells and use the normal cellular protein synthesizing machinery to translate the viral gene into viral proteins. Endogenous antigens also may include those uniquely produced by cancer cells. When many cells undergo malignant change, they begin producing unique proteins that are specific to cancer cells and are presented as foreign antigens on the cell surface. Exogenous and endogenous antigens are preferentially presented by different classes of MHC molecules. Class I MHC molecules generally present endogenous antigens, while Class II molecules prefer exogenous antigens. Because Class I MHC molecules are expressed on all cells except red blood cells, any change in that cell attributable to viral infection or malignancy may result in foreign antigen being presented by MHC class I. Class II MHC molecules are co-expressed with MHC class I molecules on a more limited number of cells that have APC function, including macrophages, dendritic cells, B lymphocytes, activated T lymphocytes, and some endothelial cells. Thus, the term antigen processing relates to the process by which exogenous and endogenous antigens are linked with the appropriate MHC molecules. Endogenous antigens are usually components of proteins synthesized in the cytosol. They are degraded into small peptides in the cytosol by proteasomes and transported by TAP, transporter associated with antigen processing proteins. TAP1 and TAP2 into the endoplasmic reticulum, where MHC class I and class II molecules are assembled. The class I MHC molecules have open antigen binding sites, so that antigen, the class I MHC alpha chain, and beta microglobulin molecule form a stable complex that is transported through the Golgi apparatus to the plasma membrane. The antigenic peptides presented by class I MHC molecules are usually very small, 8 to 10 amino acids in length. MHC class II molecules are also assembled in the endoplasmic reticulum, but do not bind with endogenous antigens because the antigen binding site is blocked by a small protein called invariant chain. Exogenous antigens are internalized by phagocytosis and small antigenic molecules produced by digestion in the lysosomes. The MHC class II complexes of the class II alpha and beta chains with invariant chain are transported to the lysosome where the invariant chain is digested and replaced by antigenic molecules that are usually slightly larger. Thus, they are in excess of 12 amino acids in length than those presented by the MHC class I molecule. CD1 is a third antigen-presenting molecule that presents a variety of lipid-containing antigens that are usually derived from phagocytosis and digestion of infectious microorganisms with very high lipid content in their cell membranes. Therefore, CD1 complexes with antigens in the lysosomes in a fashion similar to MHC class II. The pocket that holds antigen for presentation by CD1 is generally more narrow and deeper than that described of MHC molecules, and it is lined with many hydrophobic amino acids that interact with lipid, which is why the lipid is the primary source for CD1s. They are hydrophobic. but they like the fat. T helper lymphocytes. The formation of a robust immune response depends on generation of spectrum of highly specialized effector cells, or Th cells, a group of immunocompetent CD4 plus cells. The Th precursor cells responds to antigen presentation by undergoing proliferation and maturation into a phenotypical diverse population of cells that will 1. Help immunocompetent CD8 T cells differentiate into Tc cells and T memory cells, or Th1 cells, and 2. Help immunocompetent B cells differentiate into plasma cells and B memory cells, or Th2 cells. 3. Increase the capacity of phagocytosis to defend against infection with chronic microorganisms that are relatively resistant to normal levels of inflammation, so organisms that cause tuberculosis or leprosy. Th17 cells, and finally, number 4, limit the immune response to control excessive damage to normal tissue, Treg cells. These subpopulations of Th cells are critical to most immune responses, and a variety of major Th cells defects that leads to severely diminished immune responses are discussed in Chapter 9. So the APC-Th cooperation cells that are destined to become Th cells emerge from the thymus with characteristic cell surface markers. They have a function alpha-beta TCR complex and express the surface molecule CD4 and lack CD8. These are generally referred to as precursor Th cells and sometimes THP cells. 3 critical and concurrent signals between APC and THP cells are necessary to initiate the Th cell differentiation process. The first is an antigen-specific signal. The APC presents antigen held by the polymorphic regions alpha-1 and beta-1 of the alpha and beta chains of MHC class II molecules. The antigen also binds to the TCR complex of the Th cell. The strength of the intercellular antigen binding is increased by CD4 on the Th cell, which binds to a non-polymorphic region of amino acids shared by all MHC class II molecules, sequence of the beta-2 region of the MHC class II molecule. As a result of collaborative binding of the TCR to the MHC class II antigen complex, the cytoplasmic portion of the CD3, the TCR signaling complex, and the CD4 molecule interact and initiate a series of enzymatic interactions that send a signal to the nucleus. Whew, that is a lot. A second co-stimulator signal results from the interaction of a variety of adhesion molecules. The antigenic signal alone is inadequate and may even inactivate the Th cell if co-stimulatory signals are not present. The most critical interaction between B7 on the APC and CD28 on the Th cell, which activates the enzymatic activity of the cytoplasmic portion of CD28, and the initiation of additional intracellular signaling pathways. The third signal occurs through Th cell cytokine receptors. In early stages, the Th cell differentiation interleukin-1 secreted by the APC provides this signal through the interleukin-1 receptor on the Th cell. Initially, the Th cell responds by producing the cytokine interleukin-2 and upregulates of interleukin-2 receptors. Interleukin-2 is secreted and acts in an autocrine, self-stimulating fashion to induce further maturation and proliferation of the Th cell. Without interleukin-2 production, the Th cell cannot efficiently mature into a functional helper cell. So we need that interleukin-2 to help maturation. Got it. the TH subsets. Just looking at all those words. So many words to tell us what a subset means. Okay, at this point, and depending on the predominant cytokines in the immediate environment, TH cells undergo differentiation into one of several subsets. We've got Th1, Th2, Th17, or Treg cells. These subsets have different primary functions. Th1 cells preferentially provide help in developing TC cells, or cell-mediated immunity, whereas Th2 cells provide help for developing B cells, or humoral immunity. All right. And then we have Th17 cells, are cytokine secreting cells that activate macrophages and Treg cells, limit the immune response. No. And Treg cells limit the immune response. These functions are discussed in the section of cellular interaction. The Th subsets are cytokine secreting or lymphokine secreting T cells, but differ considerably in the spectrum of molecules they produce. They're cytokine receptors and intra or intercellular adhesion molecules. Th1 cells produce interleukin-2, tumor necrosis factor beta, and interferon gamma. Th2 cells produce interleukin-4, 5, 6, and 13. And Th17 cells produce. Th1 and Th2 cells have different cytokine receptors and may suppress each other. Wow. Th1 cell, what is it? Interferon gamma will inhibit development of Th2 cells, and Th2 cells, interleukin-4, will inhibit the development of Th1 cells, so they cancel each other out if need be, so that the immune response may favor either antibody formation with suppression of cell-mediated response, or the opposite. For example, antigens derived from viral and bacterial pathogens and those derived from cancer cells seem to induce a greater number of Th1 cells relative to Th2 cells, whereas antigen derived from multicellular parasites and allergens may result in production of more Th2 cells. Many antigens, tetanus virus, however, will produce excellent humoral and cell-mediated response simultaneously. How a Th cell is guided into becoming a Th1, Th2, or Th17 cell is not fully known. Some evidence indicates that different subpopulations of APC influence the choice by secreting different profiles of cytokines that may favor one route over differentiation over another. Whew. Our body works in mysterious ways. B-cell clonal selection, the humoral immune response. When an immunocompetent B-cell encounters an antigen for the first time, only those cells with specific B-cell regulations complementary to that antigen determinant sites are stimulated to proliferate and differentiate via clonal selection, resulting in multiple copies of that particular B-cell. The differentiated B-cell becomes a plasma cell and can be found in the blood. Secondary lymphoid organs, primarily the spleen and the lymph nodes, and some inflammatory sites. Each plasma cell is a factory for antibody production and is dedicated to the secretion of a single class or subclass of antibody with one variable region and therefore specificity against one antigenic determinant. One moment. Back to work. The B cell clonal selection, the humoral immune response. When an immunocompetent B cell encounters an antigen for the first time, nope, I read that. In which case, we skip down to primary and secondary responses. The immune response to antigenic challenges has classically been divided into two phases: primary and secondary response. These phases occur for development of both humoral and cellular immunity, but can most easily be demonstrated by serologic tests that measure plasma concentration of antibody over time. On initial or primary exposure to most antigens, there is a latent period, or lag phase. After approximately 5 to 7 days, IgM, antibody specific for that antigen, can be detected in the circulation, followed by the production of IgG against the same antigen. The quantity of IgG produced may be about equal to or less than the amount of IgM production. The amount of antibody in a serum sample is frequently referred to as a titer. A higher titer indicates more antibodies. This is the primary immune response. The lag phase is a result of the time necessary for clonal selection, including processing and presentation of antigens, induction of Th cells, interactions between immunocompetent B cells and Th cells, and maturation and proliferation of the B cells into plasma cells and memory cells. If no further exposure to the antigen occurs, the circulating antibody is catabolized or broken down, and measurable quantities fall. The individual's immune system, however, has been primed. The second challenge by the same antigen results in the secondary immune response, or anamnestic, which is characterized by a very rapid production of a large amount of antibody than that produced by the primary response. The rapidity of the secondary immune response is the result of the presence of memory cells that require little further differentiation into plasma cells. IgM may be transiently produced in the secondary response, and the quantity may be about the same as that produced in the primary response. IgG production is increased considerably, making it the predominant antibody class of the secondary response. It is often present in concentrations several-fold larger than those of IgM, and levels of circulating IgG specific for that antigen may remain elevated for an extended period of time. Natural infection, so like the rubella virus, may result in measurable levels of protective IgG for the life of the individual. Some vaccines, such as polio, also may produce extremely long-lived protection, although most vaccines require boosters at specific intervals. The existence of a prolonged and protective secondary immune response explains how vaccinations provide protection against certain pathogenic microorganisms. Edward Jenner, an English physician of the late 18th century, performed the first well-documented vaccine trial. Although some of the stories about Jenner's experiments are fanciful, it is known that Jenner recognized that milkmaids were protected from the deadly smallpox virus if they had previously developed cowpox, a bovine equivalent of smallpox that causes only mild disease in humans. Jenner took material from a cowpox pustule on the hand of an infected milkmaid and injected it into the arm of an 8-year-old boy. After the boy's initial inflammatory reaction to the injection subsided, Jenner injected the boy again, this time with material from a smallpox pustule. Unfortunately, the experiment was a success because Jenner is reported to have injected smallpox virus into the boy at least 20 times without the child becoming ill. In Jenner's experiment, the antigen on the cowpox virus and the smallpox virus were sufficiently similar that the cowpox antigen functioned as an altered or attenuated smallpox antigen. The antibodies and lymphocyte that recognized and destroyed cowpox also were able to recognize the smallpox virus, thereby protecting the immunized child against smallpox. In 1798, Jenner used the term vaccination, vac a cow, to describe his technique. Oh boy, and there you have some history. Injecting small boys with cow vaccines. Honestly, who remembers cowpox? I have never even heard of that. So that's hilarious. So, a class switch. A major component of maturation is antibody class switch, the process that results in the change in antibody production from one class to another during the primary immune response. The immunocompetent B cell produces MIGM and MIGD before clonal selection. After encountering antigens, however, each B cell has the option of changing the class of antibody to a secreted form of one of the four IgG subclasses, one of the two IgA subclasses, or IgE, or continue the production of IgM, but changing to a secreted form, usually a pentamer. This process is called class or isotype switch. During this process, the variable region of the antibody heavy chain is conserved, and the light chain remains unchanged from that used of the beta cell R. Therefore, the antigenic specificity of new classes of antibody also remain unchanged. The mechanism of class switch involves around of somatic recombination, during which the VDJ region encoded in the heavy chain's variable region is moved to another site of the DNA that is adjacent to the gene for a different constant region. This step is under the control of activation-induced cytosine deaminase. The intervening DNA is cut and mended with the removal of the DNA that was between the VDJ site and the new constant region. Specific recognition sites precede each constant region, and the particular constant region chosen for class switch appears to be, at least partially, under the control of the specific Th2 cytokines. For instance, interleukin-4 and interleukin-13 appear to preferentially stimulate switching to IgE secretion. The transforming growth factor beta and interleukin-5 appear to play major roles in the class switch to IgA secretions. Thus, during clonal selection, a B cell may produce a population of plasma cells that are capable of producing many different classes and subclasses of antibodies against the same antigen. Which I think is a good thing, because I think it takes the variety of cells to help fight an infection, and it's not just one specific cell, because if the body becomes immune to one specific cell type, then we may not be able to properly defend ourselves in the future once there is that switch. If there's, the body's immune, or not the body, but if the infection switches, then there is that change. So, interesting. Okay. A few antigens can bypass the need for Th cells and can directly stimulate B cell maturation and proliferation. These are called T cell independent antigens. They are mostly bacterial products that are large and are likely to have repeating antigenic determinants, multiple identical antigens determinant sites that bind and cross-link several B cell receptors. The accumulated intracellular signal is adequate to induce differentiation to a plasma cell, but is not adequate to induce class switch. The CD40 or CD40L interaction is a necessary component of the signal that leads to class switch. T-cell independent antigens usually induce a relatively pure IgM primary and secondary immune response. During the differentiation of B cells into plasma cells, the CDR portion of the antibody variable region are prone to somatic point. Mutations that lead to changes in single amino acids. Some of these changes produce better antibodies and bind more strongly to the antigen. The presence of antigen created a positive selective pressure towards developing B cells that expressed a higher affinity antibody, which results in a process called affinity maturation and an improved quality of circulating antibody over time. Plasma cells may migrate to special regions of the spleen, lymph nodes, mucosal associated lymphoid tissue that support the plasma cell's long-term survival and function, so that an adequate level of protective antibody may be available in the circulation for decades after vaccination or resolution of infection. During the clonal selection process, B cells differentiate into antibody-producing plasma cells and into a sort of long-lived memory cells. Memory cells remain inactive until subsequent exposure to the same antigen. Upon re-exposure, these memory cells do not require much further differentiation and will therefore differentiate rapidly into new plasma cells and produce larger amounts of antibody, a secondary immune response. The T-cell activation, the cellular immune response. The clonal selection process produces several subsets of effector T cells. Different phenotypes of Th cells have been discussed in this section in Th subsets. Clonal selection of other T cell phenotypes is dependent on the help by the Th1 cell phenotype. Other effector cells include Tc cells that attack and destroy cells expressing antigens of intracellular endogenous origins, viruses, infected cells, cancer cells, T regulatory cells or Treg cells, and that limit suppress the immune response, and T memory cells that provide a rapid cell-mediated immune response or reaction to repeated exposure to the same antigen. Ooh, super antigens. We're going to skip over this little section here and hit up super antigens because they are in my little workbook. Several pathogenic viruses and bacteria manipulate the normal interaction between APC and Th1 cells to the detriment of the individual and the benefit of a microbe. A group of microbial molecules called superantigens bind to a portion of TCR outside its normal antigen-specific binding site, usually the V region of the beta chain. So, what exactly is a superantigen? A group of microbial molecules. All right, and they bind to a portion of the TCR or T cell outside of its normal antigen-specific binding site and to MHC class II molecules outside of their antigen-presenting sites, either the alpha or the beta chain, depending on other particular SAGs. These molecules are not digested and processed by the APC to be presented to an immune cell. Binding results in adherence to the TCR and MHC class II molecules. Independent of antigen recognition and provides a signal for Th cell activation or proliferation. Other SAGs may bind to costimulatory molecules such as CD28 and induce a similar polyclonal Th cell activation. So, a superantigen is a molecule that activates many Th2 cell receptors regardless of their antigen specificity and binding in an unusual location. Does that sound about right? The major history. The normal antigen-specific recognition between Th cells and APCLs results in activation of relatively few cells, only those cells with specific TCRs against that antigen. Superantigens induce polyclonal THC activation regardless of the antigen specificity of the SAG-activated T cells overproduce cytokines, especially interferon, whatever that is, which is an activator of the phenotype macrophage. Extensive 1 macrophage activation leads to the secretion of interleukin-1, 6, and the tumor necrosis factor alpha, which is a primary mediator. So yes, I have a feeling that is the correct answer. Which leads us with plasma cells being antibody-producing cells. Alright, the overproduction of inflammatory cytokines result in symptoms of the systemic inflammatory reaction, including fever, low blood pressure, and potentially fatal shock. Some examples of SAGs are bacterial toxins produced by Staphylococcus aureus and Streptococcus pyogenes, including the super antigens that cause toxic shock syndrome and food poisoning. Right. I think this is where we're going to cut it off, though. Looking at my required reading, we have come to our end. There is a little bit more, but I'm going to skip over this to jump to the next chapter. There's a lot to read, and unfortunately, I have to use my time wisely, so we'll get back to the next chapter.